Okay. So we've been discussing a lot of different types of lipids, including triglycerides and phospholipids. But now we'll discuss an important application of those lipids, and that would be their use in biological membranes. That all cells, animal cells, plant cells, fungi cells, bacterial cells, all cells are surrounded by a membrane. And plant or animal or fungi cells, eukaryotic cells, cells that have a nucleus, they have membrane-bound components inside the cell. So I'll draw a cell, I'll draw the nucleus, I'll draw some other membrane-bound organelles inside the cell. And each and every one of these membranes has the same basic structure. It's made up of two layers of lipids. And so we call that the the membrane bilayer, or the lipid bilayer. And if you remember earlier when we were discussing phospholipids, I mentioned they have a tail group and a head group. And the head group of the phospholipid, which I'm representing by these circles, is hydrophilic. And so it likes being exposed to water. While the tail group, which I'm representing with these lines, is made up of the fatty acids, and it's hydrophobic. And so it tries to stay away from water. And we have two layers, one facing to the upper surface and the other facing to the lower surface. And in reality, there's no space in between the layers. They touch each other. I'm just not very good at drawing it. And the two layers of these two layers of lipids form all the membranes found in the cells, and they contain phospholipids, mostly glycolipids, which include the cerebrosides and gangliosides. Animal cells will contain cholesterol. And so the fatty acids that make up the phospholipids and the glycolipids, they're oftentimes unsaturated. And so that sort of makes because they're unsaturated, they have this sort of J shape. And so even though I've drawn them as straight lines here, they're more of this J shape, so they don't pack those fatty acid tails together very efficiently. And so that makes these membranes kind of liquid. And so a phospholipid is free to move around inside of that membrane. It can move back and forth, but it cannot move up and down because the fatty acid tails don't like being exposed to water. If we pulled one of these up into the water layer around the membrane, then it would want to move back down into the hydrophobic membrane layer so that the fatty acid tails aren't exposed to water. But anyway, one of the interesting features of cells is that they can adjust the rigidity of their membranes by changing the saturation level of the fatty acid tails. That if the temperature gets hotter, then the lipids are more able to move around. And so in order to control how they move around, the cells would produce more saturated fatty acids and saturated phospholipids than unsaturated phospholipids. And so that controls the rigidity of the membrane. Another way that animal cells in particular will control the rigidity of the membrane is by including cholesterol, which, see if I can draw a tiny cholesterol. And so the cholesterol, because it has a very rigid structure, if you have a lot of cholesterol in the membrane, it will make those membranes more rigid. Now, another important component of these membrane bilayers are proteins. So I'll draw in a protein. We won't worry too much about what it does at this point. And we'll extend our lipid bilayer out a little further. So that we have these proteins suspended in this sea of lipids, and we call this a fluid mosaic model. And we call it fluid because these different components, they aren't frozen in place. They're able to move around to some extent inside of that membrane. But they can't really escape the membrane because they're too hydrophobic. They don't like the water on either side of the membrane. But anyway, small molecules like water, oxygen, carbon dioxide, they can pass right through the membrane fairly easily. So I'll draw water. Because it's a small molecule, it can move through the membrane without too much trouble. And that movement of water across the membrane surface leads to osmosis, which we discussed in an earlier chapter before the online classes. However, larger molecules like saccharides, sugars, they can't move through the membrane very easily. And so in order to get those molecules through the membrane, we need a special type of protein called a transporter protein, or a channel protein. And they have a tube inside the protein that allows, let's say, a sodium ion to move through that tube. And there are two different types of these transporter proteins. Well, actually, there are hundreds of different types of transporter proteins, but generally they fall into two classes, one that requires energy and one that doesn't. So facilitated diffusion is simply that the protein just opens up a hole in the membrane and the molecules can move through based on their concentration. If we have a high concentration of sodium ions inside the cell, then if we open up a hole, those sodium ions will rush out to the outside of the cell. Or vice versa. We always go from high concentration to low concentration. So I'll say high to low, but it does not cost energy. Whereas active transport does the opposite. That active transport grabs a molecule on one side the protein would sort of bind that molecule on one side of the cell and pull it inside. And so it goes from low concentration to high concentration, and it requires energy. So the cell has to spend energy to pull molecules through the membrane when it uses active transport. But if it's just using facilitated diffusion, then it does not require energy, and the molecules and ions can flow back and forth depending on the concentration on one side of the membrane versus the other side of the membrane. So hopefully that makes sense, that there's a lot more we could discuss on membranes and membrane proteins and how things move across. It gets very complicated very quickly, but I think this is an okay introduction to the concept of cellular membranes.